So next, protoplasm theory. The protoplasm theory developed by Hartwig. First one, Dujardin. Second one, Parkanji. And third one, Hartwig. Dujardin called the living matter as sarcode. Who given the name living matter? First time, Alfonso Corti. So Alfonso Corti called the living matter inside the cell. For that sarcode name given by Dujardin. For that sarcode protoplasm name given by Parkanji. And protoplasm theory. So protoplasm theory given by this Hartwig. And plant cell without the cell wall. Plant cell means it is with cell wall. If we are removed cell wall, without cell wall, the plant cell is called protoplast. So here another word, protoplast. Protoplast is called plant cell without cell wall. Plant cell or any cell without cell wall is called protoplast. So protoplast, the term coined by Hans Steen. First time Hans Steen proposed the word that is called protoplast, the word given by Hans Steen. Hans Steen proposed the term protoplast. What is protoplast? Any plant cell without cell wall is called protoplast. Protoplasm theory given by Hartwig. Protoplasm, the word given by that living matter Parkanji, and living matter word given by Alfonso Corti. For that living matter, Dujardin called as sarcode. So these are the terminologies you have to remember. Repeat the video a number of times, and then with according to the year, chronological year. So year is important. According to year, and you remember a scientist and his contribution. And after the Hans Steen discovery, Knoll and Ruska invented electron microscope, and Knoll and Ruska are credited with inventing the first electron microscope in 1931 at the Technical University of Berlin. So first time Knoll and Ruska. Knoll and Ruska in the year 1931 discovered important electron microscope. First time they discovered electron microscope. Fritz Zernike invented phase contrast microscope. Using his one can observe without staining. So what is meaning of a phase contrast microscope? Without staining you can observe any cell. But in the compound microscope, electron microscope, any microscope, we have to use a dye, a stain to observe outside parts or internal parts of a cell. But Zernike first time discovered a microscope that's called phase contrast microscope. By using this microscope, you can observe any cell without using a staining. Okay, Zernike. Phase contrast. microscope. So this is a discovery of the microscope. Zacharias Johnson and Francis Johnson discovered first useful compound microscope. Then Robert Hooke discovered cell. Anthony von Leeuwenhoek first time discovered living cells by using his simple microscope. Noll and Ruska discovered electron microscope. And Fritz Jernik discovered phase contrast microscope, one of the microscope without using any staining or dye we can observe plant cell or animal cell. So one of wonderful discovery, phase contrast microscope. So up to now this is called the history or introduction of cell biology. After the discovery and introduction of cell biology, now look here, every living organism is made up of cells. So this is a cell. In the middle, this is the nucleus. Within the nucleus, there are DNA, also called chromosomes. This is one part. Coming to here, the outermost covering of the cell is called cell membrane. Another covering is there. This is called a nuclear membrane. Inside the liquid, this liquid is called cytoplasm. Okay, within the cytoplasm there are cell organelles. Coming to DNA creates DNA in the mitosis, that is called replication. So DNA creates another DNA, that is called replication. DNA creates mRNA. This is called transcription. This mRNA prepares proteins. This is called translation. So replication, transcription, translation. This is one part going on in the cell, in the middle, in the nucleus. And coming to the outside of the cell is called cell membrane. And surrounding the nucleus, the membrane is called nuclear membrane. And inside the liquid in the cell is called cytoplasm. Within the cytoplasm, cell organelles are present. So one by one we discuss in briefly now. The first topic is now cell membrane. So first part, cell membrane. So cell membrane is the outermost covering of every cell or animal cell or plant cell. Plant cell or animal cell or protoplast, whatever you take, it is surrounded by a cell membrane. The term plasma membrane, see, the another synonymous word to the cell membrane is plasma membrane also. So the term cell membrane given by two scientists, Nageli and Kramer, coined the term cell membrane. And plasma membrane, this terminology, the term given by Plove. So Plove given the new term plasma membrane, cell membrane term given by Nageli and Kramer. Ernest Overton given an idea, that is cell membrane is made up of lipids. Ernest Overton, most important. Ernest Overton given an idea, cell membrane made up of lipids. Most important idea given by this Overton, Ernest Overton. Already we know that cell membrane is made up of phospholipids. But first time this information given by Ernest Overton. Ernest Overton hypothesis stated that cell membranes are primarily composed of lipids. This is a statement given by Overton. In the 1925, most important 1925, Ernest Gorter and Grendel extracted lipids from the RBC. Gorter and Grendel. Look here. Gorter and Grendel. They extracted lipids from the cell membrane from RBC. And they coined that these lipids are present in the form of bilayers. They are present in the bilayer form. So 2 layers are present. So in the cell membrane, lipids are present. The idea given by a scientist. After that Overton's information, Gorter and Grendel extracted the lipid components in the RBC cell membrane, and they observed these lipids are in the double layer form, and they proposed a model of a cell membrane that is called lipid bilayer membrane structure. So cell membrane is in the form of a lipid bilayer. This theory given by Gorter and Grendel. First time Gorter and Grendel extracted lipids from the cell membrane of RBC, and they observed layer 1 and layer 2. 2 layers they observed in this lipids, and then they proposed lipid bilayer structure of a cell membrane. In this lipid bilayer structure, what they observed, the outside, both the outsides, they are called polars. Polar heads are there. They are called hydrophilic heads. These heads are with lecithin and cephalin. They observed that these are the hydrophilic in nature, and they are made up of lecithin. And second one, cephalin. These are the lipids. And in the middle, what they observed, they are hydrophobic, and they are made up of cholesterol. See, cholesterol. Lipid bilayer structure given by Gorter and Grendel. They observed the lipid bilayer structure in the RBC cell membrane, and they proposed lipid bilayer structure to the cell membrane. He also observed that is outside heads are present. In these heads, lecithin and cephalin, the lipids are present. They are hydrophilic polar ends. In the middle, there are hydrophobic in nature and is made up of cholesterol. So this is the information given by this Gorter and Grendel.